For the last 3 years, biology has been among the most failed subjects at O&A level, yet it is a vital subject in the health sector. Having realized this concern, I opened up a YouTube channel which I identified as Easy Science International to assist both O&A level students understand biology better, with an aim of elucidating passion for science among learners, especially in this trying moment of COVID-19. This idea was conceived in my second year at MUST, but it was not implemented until in this COVID-19 lockdown, when one of my OBs requested me to assist a sister in A-level biology. I found it difficult to assist her since she was far apart from me and the alternatives were expensive due to data and airtime expenses. Easy Science International is a platform dedicated to helping you achieve your dreams in the world of academia.
Yes. Welcome to Easy Science International and greetings from Ivan. We want to send our sincere thanks to our audience in Uganda, Kenya, United States of America, Tanzania, and all other countries. Stay glued to Easy Science International and there for more. So, we are looking at the ANGELMAN'S experiment. We've looked at the procedure and the control, the procedure of the experiment and the control experiment. Now, we want to see what do you observe when you expose cladophora, which is in a drop of water, and you avail motile aerobic bacteria, and you expose it to light of varying wavelength, and another slide you expose it to total darkness. We want to make observations, and here we make 4 observations. Let's go. Observations with their deductions. Now, observation number 1. Motile bacteria cluster near the filaments in the region of blue light, that is 400 nanometers, and red light, which is 650 nanometers or 700. So we are seeing that our first observation, when you look at this cladophora under a microscope, and we are looking at the real experiment, not the control, we see that the motile bacteria, which are aerobic, cluster. They make clusters, that is in groups, near the filaments in the region of blue light and red light. What do we deduce from here? Deduction number 1. Since the distribution of aerobic bacteria depends on the concentration of oxygen, and oxygen being a byproduct of photosynthesis, remember the equation of photosynthesis? So we see that oxygen is given off in the process, and the very oxygen is required by the aerobic bacteria for their survival. So we expect the aerobic bacteria to be abundant in the region where we have a higher concentration of oxygen along the cladophora thread or the green algae thread. So we see then the red and blue light are the most effective for photosynthesis. Yes, so are seeing the motile aerobic bacteria being attracted to cluster around the red and blue regions. We deduce that these regions of light, or the wavelength of 450 nanometers blue and 650 nanometers red, are the most effective wavelength in stimulating the process of photosynthesis. In the result, they enable the cladophora to carry out photosynthesis at a higher rate in those regions, releasing oxygen, which oxygen attracts the aerobic motile bacteria in a process called chemotaxis. So they can be attracted to regions with higher concentrations of oxygen. So that is the first observation and the first deduction. Observation 2. Motile bacteria cluster around the edge. They cluster around the edge of the cells adjacent to the chloroplasts. We observe in this experiment, remember, green algae is comprised of spiral chloroplasts when you look at its structure. Now, we see that the motile bacteria cluster around the cells adjacent to the chloroplasts. Why? Deduction 2. Oxygen is more concentrated near the chloroplasts. This shows that the chloroplasts are the sites for photosynthesis. So, since oxygen is highly abundant in high concentrations around the chloroplasts, this insinuates that true, the chloroplast is the site for the process of photosynthesis, not anywhere else. Yes. So far, that's our second observation and second deduction. Remember, we are looking at the ANGELMAN'S experiment. The experiment which clarifies that indeed red light and blue light are the most effective lights in stimulating the process of photosynthesis. Now, so far we've proved that, and we also proved that indeed the process of photosynthesis occurs along the chloroplasts. As in, it is the chloroplast which is the site for the process of photosynthesis. That's why the motile bacteria are attracted to the cells adjacent to the chloroplast. Now, let's look at the second last observation. So we are looking at observation 3, our second last observation. Now we want to look at our control experiment. Remember, the second slide containing the drop of water with our cladophora or green algae thread was placed in total darkness. So we want to see what really happened with the motile bacteria, which are aerobic in nature. Observation 3. The aerobic bacteria of the slide in the dark are immobile. Immobile meaning that they don't move. In other words, stationary. But later when the slide is placed in light, the bacteria cluster around the algal filaments. Deduction. What do we deduce from this observation? Darkness prevents the process of photosynthesis because we've seen that for photosynthesis to take place, there must be light. So, preventing the process of photosynthesis implies that even the byproduct of this process is not produced, and that is oxygen. Motile bacteria being attracted to oxygen concentrations means that they won't be attracted to anywhere, so they remain immobile. So what really happens? Darkness prevents photosynthesis, which stops evolution of oxygen, creating anaerobic conditions which do not favor aerobic bacteria activity. Now, anaerobic conditions in the sense that there is no oxygen. Since our bacteria are not anaerobic in nature, being aerobic, they won't move. Their activity will be low because respiration is very limited. And hence they can't move up and down. They can't get that energy to move up and down and make a lot of that to and fro movement. So they remain immobile simply because there is no oxygen. The third observation is that. Then let's look at the last observation, and that is observation 4. There is hardly any bacteria in the UV, green, and infrared regions of the spectrum. There is hardly any bacteria found there. So hardly means that you might find there one, but not in a cluster. The deduction here. So deduction 4. UV light, green light, and infrared light is hardly trapped. Can use the word trapped. Is hardly trapped by chlorophylls, hence least used in the process of photosynthesis, with no or little evolution of oxygen. Yes, this light which you cannot see with your naked eyes, infrared light, I told you to tell me the color of this light. Just put that color in the comment box. We shall harmonize on it. So, and the UV light, ultraviolet light, that one you can also not see it with your naked eyes. Then green light are not trapped by chlorophylls. So remember, in physics, you see what the object reflects. So if you are able to see that this lid of this marker is green, it means that this green object or marker, it means that this absorbs all other colors and reflects green. So even the leaves appearing green, it means that they absorb most of the colors and then they reflect green. Basically the chloroplasts, which are having what we call chlorophyll. So chlorophyll will absorb other colors and reflect green. That's why it appears green. Now, on top of reflecting green, there are other colors like infrared, which are not trapped. They just pass through the leaf. Then there is also UV light. It is also not utilized by the chlorophyll, so it is just left to go away. It is not bounced back or refracted. It just penetrates the leaf and goes. So, since these illuminations do not stimulate the process of photosynthesis, we don't expect a byproduct of photosynthesis, and that is oxygen, to be released. And therefore, in a region or where you expose this kind of light, infrared light, green light, and UV light, you won't expect photosynthesis to take place at that segment of the cladophora or green algae, and therefore no motile bacteria found there, aerobic bacteria, all of them won't be found there, or you will find there one which will be there by mistake. kumbaga tekula musiuvu. That on an occasion you can find somebody who's not ready for that occasion. So this bacteria might be confused in that way and finds its way there. But you won't find the majority. That is, in a cluster, you will never find them there because the conditions are intolerable. Yes. So those are the 4 observations we make. Now we want to summarize them on an illustration. So this is a simple illustration of what we've been talking about. We have a thread, which is cladophora, put on a microscope slide, this one, as you can see, and this slide, you put this thread in a drop of water on a microscope slide. You expose the thread to motile aerobic bacteria, which are these ones. Now, the first slide, you expose it to light of varying wavelength, and this is in summary what we've observed. We observed that aerobic bacteria cluster in regions of blue light mostly. Then we can also find them in indigo and violet, because this region, depending on our action spectrum and basically the action spectrum and the absorption spectrum, we see that the blue to violet region stimulates the stimulates effectively the process of photosynthesis. And they also cluster in another region, which is the red region. When you expose the red light, you find that there is another cluster of aerobic bacteria. Then we also saw that in the green, infrared, and UV light regions, we hardly find there any motile bacteria, which is aerobic. So you hardly find there any. So you might find there 1 or 2. The same applies to orange and yellow, because they hardly stimulate the process of photosynthesis, and you won't find their oxygen. So these motile bacteria are chemotaxic. They are attracted to areas with higher concentrations of oxygen to support their aerobic activity. So we expect that here and here. Reason being, along this region, that this segment, this cladophora is able to carry out photosynthesis and hence release oxygen, which gets higher and higher in concentration around here, and hence attracts these chemotaxic mobile aerobic bacteria to cluster around there. The same applies to this region. However, here you find there a few, because even when you look at our action spectrum, it is not zero. Yes, some portion of it is absorbed, but not to that higher level. Then here you will find there none, and here, because these kinds of light are invisible and not absorbed at all. So our infrared light appears like that. Kindly comment about the color you are viewing in the comment box below. So basically, this is what happens. In total darkness, you will find that these bacteria are just randomly distributed along the slide. Their distribution is independent of the color of the light, or the type or the wavelength of light being exposed to. Why? They are in darkness, and in darkness we don't expect their light. Yes. So here we don't expect their light, so they are the cladophora or green algae thread is not carrying out photosynthesis, that is along the chloroplast. So we also saw that the mobile aerobic bacteria cluster around the chloroplast, cells which are around the chloroplast. So, and that shows that indeed, with this we can't view on this illustration that you see with your eyes and a microscope. So we see that being close to cells with which are near to the chloroplast, it means that indeed the chloroplast is the site for carrying out photosynthesis, and hence it has a higher concentration of oxygen than the edges of the green algae. So basically, that is the experiment which was put forward by a man known as Angelman, and we named it after him. It shows us how different wavelengths of light stimulate the process of photosynthesis. So he used motile bacteria because these bacteria being aerobic, they will be attracted to oxygen. So he can't see oxygen with his naked eyes. The way you can't see infrared light with your naked eyes, we can't see oxygen. There is oxygen here, there is carbon dioxide between me and the camera, but I can't see them. Now, I use an indicator because when I know that there is this one, I must know that this one is there. Have you ever gotten a friend in class and even teachers identify you because of that friend? When they see him or her, they say that Nachimwero is here, so Sechizivu must be coming. So I expect if Nachimwero is invisible and as he sees Sechizivu, I expect Nachimwero to be very, very close around. So when I expose this aerobic bacteria and I see it being interested at a certain corner or at a certain segment of a thread, I just detected that there is oxygen because the two move together. So this is the idea which Angelman used. So he exposed it to light of different wavelength and he observed the 4 observations we did and the scientific explanations we've tried to give them. So thank you for watching. Stay glued to Easy Science International. Always don't forget to subscribe. We just thank you for those who have subscribed. Encourage more people to subscribe to our channel. Next time we meet, we are going to be handling the biochemistry of photosynthesis. This is very interesting. They are not to miss. Serena Javen is my name. Have a blessed day.